I feel kind of like a spotted owl at a loggers convention because I work with a small, wimpy, nondescript, easily overlooked, non-biting mosquito. Really, what the talk is about is, can we look into the past and sometimes the present, and can this tell us something about tomorrow? And it's really perspectives from phylogenetics, genomics, and development in the pitcher plant mosquito. It's a joint effort between Christina Holzapfel and myself. We've worked together as evolutionary partners for more years than we're willing to admit. We'd like to thank many colleagues, both in Oregon and at other universities around the world, and especially the National Science Foundation that has been extremely kind for many years. I think really to bring it home that what is the potential that could happen. This shows deaths from malaria in eastern United States. This is the main portion of our study areas, this part of the world. Chris and I are mainly interested in evolution in temperate environments. And this shows deaths from malaria in 1870, primarily along the eastern seaboard and up through the Mississippi River Valley. And you think, well, there's very few deaths from malaria there today. But the major vector of malaria is omnipresent throughout this range, Anopheles quadrimaculatus. So the potential is there. But to bring it back to climate change, that really temperature is having a direct effect on the non-biological systems of the Earth. This is a picture of the Muir Glacier in 1941 and 2004, and as you can see in the intervening years, it has receded all the way up the valley. This is due to the direct effects of temperature melting ice. But as we like to point out, animals are not glaciers. And we have to ask, what are the environmental forces that are driving the biological response to climate change? The other way I like to put it is we're beyond throwing the family cat in the microwave and yelling global warming, that there are other factors at play here. Abiotic consequences of climate change. We're getting warmer winters in particular. In eastern North America, winters are warming faster than summers. This is leading to earlier springs, and if you just saw with the ticks, we're getting early development and even winter development of the ticks, later onset of winters, and longer growing seasons. These are still abiotic aspects of climate change. So now let's look at some of the biotic consequences that have been widely documented. That there's been a general northward expansion of species ranges in the northern hemisphere, and earlier development and reproduction in the spring with a later onset of dormancy or migration in the fall. These are biotic responses to climate change. And we'll now bring in the principal actor, the pitcher plant mosquito Wyamia smithii. Here is a female laying her eggs in a pitcher plant leaf. This is the purple pitcher plant, which extends from Florida north to Labrador and west to Saskatchewan, and now even some sightings in British Columbia. The female drops her eggs from the air onto the water and the leaves. The larvae go through 4 larval instars, pupate, and regenerate the life cycle. The plant has evergreen leaves. This is an early summer plant that is in bloom. Here are last year's leaves that have persisted through winter, leaves from early spring, and leaves that are just now opening. And the mosquito completes 1 to 7 generations per year, depending upon the latitude you're looking at. This is a summer picture. In the winter, things look very different. There are evergreen leaves on the plant. The plant persists. The Wyamia overwinters as a larva in diapause. The diapause is initiated, maintained, and terminated by photoperiod. And we can ask, since temperature has been a very predominant theme in this talk, how important is temperature as an anticipatory cue as compared with light? And we have put a probe, a watchdog, in a single pitcher plant leaf, and this represents temperature inside of that pitcher plant leaf throughout a whole year, taken every 2 hours. As you can see, there is a seasonal pattern. It's warmer in the summer than it is in the winter. But there's a tremendous amount of variation from day to day, week to week, month to month. We actually know that they're going into diapause at this locality at about day 300 Julian date. Yeah, there's a little cold snap here, but it's warm before and afterwards. So temperature is a pretty fuzzy cue on which to bet your life. This is when to go into diapause is a life or death decision. We superimpose day length over this picture. We can see that day length is highly regular. In fact, the number of hours of light per day today in New Haven is the same as they were 10,000 years ago and will be 10,000 years in the future. Climate change is not affecting the rotation of the Earth about the sun. And it is not changing day length. Day length remains a highly reliable cue through evolutionary time. And if you were going to bet your life on a seasonal cue, on being able to anticipate and prepare for the changing seasons, would you bet on temperature or light? In fact, a wide variety of animals, thousands of species of animals in the temperate zone, rely upon day length to program their seasonal activities. All of these animals are photoperiodic. They use the length of day to program some aspect of their seasonal life histories, ranging from rotifers to rodents. And I guess this must be one of Hopi's animals, because here's the ear tag. How should response to day length? I said that all of the animals all of the animals on the previous slide live at temperate latitudes, and they exist over a wide variety of latitudes. How should the use of day length change with latitude? Here we have day length and Julian date of the year for a southern locality along the Gulf of Mexico and for a northern locality along the Canadian border. The decision to enter diapause or continue development is made in the late summer, early fall, and that's blown up over here. And I've plotted the onset of winter at the northern latitude and at the southern latitude. A northern animal to go and enter diapause before winter would use a switching day length or critical photoperiod around 15 3/4 hours of light per day. A southern animal would use a southern day length and enter diapause at a little over 12 hours of light per day. So the prediction is that the switching day length or critical photoperiod should increase with latitude. And in Wyomia-Smithii, this map shows our principal collecting localities. It's very nice. The pitcher plants are long-lived perennials living for a century or more. So we can go back to specific localities and harvest whatever population attributes we want over a long period of time. In fact, one of our original sites here in Florida, we've collected from the same plant for over 30 years. And what we find is when we bring them into the laboratory, grow them through several generations to minimize maternal field effects, and then determine the switching day length, we find there's an extremely tight correlation with latitude. In fact, the switching day length or critical photoperiod in Wyomia-Smithii tracks geography more closely than any other known ecogeographic trait. And we also find that it has a high heritability, all the way from 0.07 to 0.77, depending upon the population you're looking at. Okay, I've said that light is important, that unless you're a foolish organism, and there probably are some out there, Father Darwin calls them home, that you're going to rely on day length to program your seasonal development, but can we show that this really affects fitness? And the answer is we can, because here is a pitcher plant in the spring. There are no new leaves on it. All the overwintering larvae are here in the water-filled leaves of this carnivorous plant. For those of you who have a ghoulish tendency, you'll notice that all the hairs here point downwards. We bring the whole plant into the laboratory, and we can rear Wyomia-Smithii in its natural microhabitat. In fact, we run our experiments determining fitness by feeding them freeze-dried Drosophila to mimic the natural prey capture cycle of the host plant. And we are blessed with climate control rooms that are computer-driven, and we can generate any climate from the tropics to the Arctic, including light temperature and humidity. And we can, unlike nature, we can juxtapose either of those. We can give a Florida mosquito a Florida thermal year, including daily and annual temperature cycle, and give it the light cycle of Maine, just to play around with it. And we define fitness as the year-long cohort replacement rate, not just summer fitness. So we start out, we program one of these rooms for a continuous year, and we start out with an initial cohort of larvae that we put into pitcher plant leaves, and that constitutes the first generation of the first summer. The long days of summer promote continuous summer generations until the short days of fall induce a larval diapause. They overwinter as larvae in the leaves until the long days of spring stimulate resumed development, and we get the first generation of the second summer. The adults come out, lay eggs, the eggs hatch, and we get a cohort of hatchlings 15 months down the line from when we started. And fitness integrated through all 4 seasons then is the number of hatchlings after 1 year divided by the initial cohort size. This, you get a sample size of 1 every 15 months. And we now can play around with the thermal year and the photic year. Remember, we're programming daily and seasonal variation in temperature to mimic, to start with, a northern locality. We'll say, what happens to fitness of northern localities due to climate change? So we have to have a control. So we give the northern population a northern thermal year and a northern photic year, and we get a baseline level of fitness, the year-long cohort replacement rate. We now give it a southern thermal year equivalent to 180 years of climate change at its current rate. That is not imposed over evolutionary time, that's imposed instantaneously. And we leave it with its native northern photic year, and we say, what happens to fitness? It increases 47%. So for Waimea, like many other temperate organisms, as Ray Huey's lab, for other ectotherms, as Ray Huey's lab has pointed out, warmer is better. Now, we can test the effects of light because we've established that this is a thermally benign climate. What happens now if we give it the same thermally benign climate, but give it the photic year of 6 degrees further south? And the answer is an 88% loss in fitness. So that photoperiodic mistiming of seasonal development is very lethal. And it's the correct interpretation of light, not temperature, that matters. We've done some work. We're saying, okay, a lot of people working with climate change assume everything is static. But evolutionarily, everything isn't necessarily static. Is there evolutionary response? I've said this is a highly heritable trait. What can we say about the genetic architecture? Well, I could go on for that for a long time, but we'll show you some of our recent data with QTL mapping. That we've used the geographic gradient to focus on 3 populations, Maine, New Jersey, and Alabama. The Waimea spent the last glacial maximum oppressed closely to the glacial front, and within the last 13,000 years has migrated to northern Maine. We know, using this as to calibrate a molecular clock, we estimate that the Maine and Alabama populations are on the order of 200,000 years separated in evolutionary time. We drew a male and a female from these 2 populations, grew them through 7 generations in the lab to promote independent free recombination, and then genotyped on the order of 250 individuals according to their critical photoperiod, and used rad tags as markers, and developed a linkage map and then QTL map for regions of the Wyomia-Smithii genome responsible for the evolution of photoperiodic response over this gradient. This shows the log odd score, and you can think of it as the likelihood that a given region of the genome, here are Wyomia's 3 chromosomes, as an indicator that a given region of the genome, the likelihood that it contributes to the evolutionary divergence of these 2 populations. And points over this line are considered significant. Those below it are not considered significant. And what we see here is regions on the 2nd and 3rd chromosome as being scored as important in the evolution of photoperiodic response between New Jersey and Maine. We now look at Alabama versus Maine, and much to our surprise, we obtained a very different map. That this tells us that it's 3 regions on the 1st chromosome and none on the 2nd or 3rd that have been important in the evolutionary divergence between Maine and Alabama. We know that these are 3 peaks and not shoulders of 1 peak because the 2 lateral peaks are different in sign from the central one. It could be argued that what we are seeing here is not a difference between populations, but since we drew 2 individuals at random, one from each population, what we're looking at is polymorphism within populations. So we made a reciprocal cross between Alabama and Maine and looked at that QTL map, and we got something entirely different. That is, there were QTL on the 1st chromosome, but at the other end of the 1st chromosome than here. And there's one on, very prominent one on the 2nd chromosome here, which is totally missing here. So this means that the perception of the genetic basis of the trait you're looking at depends critically not only on the population within the species you're looking at, but the individuals from within that population that you choose to evaluate genetic variation. So our lab has shown that the genetic mechanisms underlying photoperiodic response are going to be far more complex than anyone expected from knowledge of other biological timing mechanisms, i.e. circadian rhythms. Genetic background, that is the genetic composition of the rest of the individual or the rest of the population, is essential in evaluating the genetic architecture underlying adaptive evolution. That there clearly is going to be gene-gene interaction, that epistasis, pleiotropy is rampant. We now ask, how do our genetic results to date help us to interpret and predict the impacts of climate change? You thought, aha, he's going to return to the topic, finally. So let us take the isotherms of eastern North America from the Gulf of Mexico to the Canadian border, where each line denotes a given temperature, mean daily temperature at that latitude and at that time of year. A couple of things emerge from this graph. First of all, one traverses very few isotherms in midsummer and a lot more isotherms in midwinter. So the climatic gradient of eastern North America is primarily one of winter cold, not summer heat. And hence my question about vernalization and chilling. And the second thing to note is, suppose we take the developmental zero of our organism at 15 degrees. We can see that the 15-degree isotherm, temperatures warmer than that are about 10.5 months long along the Gulf of Mexico and less than 3 months long along the Canadian border. So that the climatic gradient of eastern North America is not only one of winter cold, it's also one of length of the growing season. We now take as a reference point, chosen arbitrarily, we let 120 years of climate change take place, and we say this is what we expect the climate up here to look like in 120 years of climate change at its current rate. What do we notice? There has been more than a doubling of the growing season. So that selection, as Chris and I alluded to earlier, the selection is working on the timing of seasonal development, not necessarily on the direct effects of temperature, because warming winters promote an earlier onset of spring, later onset of winter, longer growing seasons, and therefore impose natural selection on the timing of development, reproduction, migration, and dormancy. How does this happen? Here is a northern locality with the same plot indicating about 15 3/4 hours of light per day. Climate warming produces later winters, which should select for a shorter, more southern-like critical photoperiod. Does this actually happen? Are the responses of populations in nature actually becoming more southern-like? And the answer is yes, they are. That we published this article in 2001. It was the first article to show actual genetic change or evolutionary response to climate change. We wouldn't have believed the data ourselves had we not actually replicated the experiments. Due to dumb, stupid, ignorant luck, we had determined critical photoperiod over geographic gradients to answer different questions. And I mean, 30 years ago, climate warming was a non-issue. So we didn't design experiments to answer that question. We designed these experiments to answer other questions. We made a collection over this gradient in 1972 and again in 1996. 96 in red, as you can see, there is a shift towards shorter, more southern-like critical photoperiods over this 24-year period. This shift is significant, and in fact, there's a year by latitude interaction, which is also significant. That means the slopes of these two lines are different. Why do we expect that? That is because climate change is taking place faster in the north than in the south. So we would expect to see a greater response in the north than in the south, and that's exactly what we see. There is another problem with what we call then and now experiments, and that is you have to be sure that you are imposing exactly the same conditions now as you did back then. And this experiment has a built-in control because we expect little or no change in the climate in Florida, which is down here. And hence, if we are recreating exactly the same conditions, these points should fall on top of each other, which they do. To answer later questions unrelated to climate change, we made collections just 5 years apart and used a much more precise measurement of critical photoperiod. Again, there is a shift towards shorter, more southern-like critical photoperiods, and there is again a significant year by latitude interaction, even with the smaller sample size. So this experiment repeats, and we see that there has been a genetic, since we grew them through 2 intervening generations, and critical photoperiod breeds true in the lab. This represents a genetic shift, actual evolution in nature, that is detectable over a 5-year period. This is evolution on the hoof at breakneck speed. Since publication of our article, other articles have come out showing genetic change in plant and animal populations. These data are hard to come by. People do not have data from 20, 30 years ago, particularly ones where they can factor out genetic from phenotypic change. All of these genetic changes involve the timing of seasonal development, or reproduction. Not a single one of them has shown a difference in thermal optimum or thermal tolerance. And need I point out that all of the bottom row here are hosts for disease vectors. That brings us to this wonderful article that we just came across, and that is the reproduction of deer, herons, and frogs, and the blood meals that are taken from those animals and whether they are in a reproductive state or not. And there is a remarkably close correlation between blood feeding of mosquitoes and the reproductive status of the hosts. That is, the mosquitoes are attacking the mother deer and fawns when they're distracted with reproductive and developmental processes. They're biting chicks in the nest, and they're biting calling frogs. So, climate change, we've shown, is affecting the seasonality of insects, birds, and mammals. It's changing the optimal time to do things. Then, for a mosquito, it is changing the optimal time of year for the mosquito to do things, based strictly on climate. We didn't introduce the host. As I said, why am I as a wimpy, non-biting mosquito? But for the rest of you in this room, if you're working at temperate latitudes, there is going to be not only selection on the optimal timing of seasonal activities of the host and optimal time for seasonal activities of the mosquito, but there's going to be host by vector interaction and the optimal time to do things. So Chris and I would like to emphasize the importance not only of host seasonal timing and what it means to the mosquitoes, but therefore on vector by host interaction and their ability to spread diseases. So today, history tells us what can happen. The contemporary distribution of vectors tells us that history has the potential to repeat itself. Ultimately, the genetic background of individuals and populations will determine their evolutionary potential to survive and reproduce in changing environments. Climate change is resulting in altered selection on seasonal timing of vectors, their hosts, and therefore vector-host interaction. Vectors may track the seasonal timing of their hosts, or hosts may shift reproduction into more vulnerable seasons. Animals are not glaciers. Light, rather than the direct effects of temperature, would become increasingly important in determining the timing of life history events as pathogens, vectors, and their hosts move into and expand in the temperate zone. And we'd like to leave you with the following grim message that all of these animals are out there in eastern North America. They're waiting. This is happening. Are we ready for them? Thank you.